This lecture is going to be done on Monday, October or November 9th, and it's a lecture on vision and hearing, looking at the processes of both those two senses. So we'll start with vision. When we talk about vision, we have to talk about what it is we're seeing. And what it is we're seeing is basically light energy. Light energy is just a small little subtle portion of the full spectrum of electromagnetic energy. That includes things like radar, gamma rays, X rays, ultraviolet light, and so on. But the part that's visible to us is that small spectrum that we would call light energy or the visible light energy. Now, when we talk about the light energy itself, we have two measurements that we can make. The first is the wavelength. So if you take a look at that image there and you see how it goes from 400 to 700, that represents the wavelength of the light. And the wavelength, if it is smaller, will be more in the blues and purples. And if it is larger, will be more in the oranges, reds, and yellows. The wavelength represents the hue or the overall color. Uh, Other species can see actually more wavelengths of light than we can, so they can see colors that we cannot comprehend. Additionally, the intensity of the light, so how strong it is, will represent its overall brightness. So, a color in low intensity will be a really, really subtle color, like, for example, a wavelength of light at around 700, but low intensity will be pink. But wavelength of 700 with high intensity will be a dark or bright red. Now, one other interesting thing about the light is when we see light, it's actually the light that's not being absorbed by objects or people or everything else. When we look at an object and we see that, let's say it's red, that means that that object is absorbing all wavelengths of light except the red wavelengths, which are bouncing off the object and then entering our eyes, where we can then perceive it as red. So in truth, it's not really red, it's actually every color but red. Now, when it is we look at things, we need to follow the path of light energy and how it goes through our eyes. The first thing it hits is our cornea. The cornea is the outer layer of the eye, along with the eyelashes, helps us protect our eyes from things like dust and dirt. Then the light will go through the pupil. The pupil is that kind of black spot in your eye that will open and close depending on the amount of light that gets into it. The pupil, if there's more light, will close, will contract, so that way the eye doesn't blind itself. And if there's less light, the pupil will open up more to allow more light in to allow us to see better. It is the iris, which is the colored muscle of the eye, that opens and closes the pupil to let light in. Then the light hits the lens, and the lens will focus the light and shine it onto the back of the eye, as well as flip the image upside down. Your brain will eventually flip the image back right side up. And the lens obviously just makes it more focused and a more clear light, eliminating some of the different inconsistencies that we might see. Eventually, the light is projected onto the back of the eye, which is a large structure in the back of the eye known as the retina. Now, in the retina, we have the fovea, and that's where the majority of the light is focused on. And you can see the fovea here, where the candle is upside down. The fovea is the central focal point of the eye, and it's where the majority of the light hits. Now, when it hits that part of the retina and other parts of the retina, it stimulates cells in the back of the retina called rods and cones. Rods and cones are what really allow us to see, because rods and cones are getting stimulated by the light, which then creates electrical signals. So this is where transduction occurs in the eye. So this is where light energy is converted into electrical signals. Rods and cones get stimulated based on the wavelengths of light that it's picking up. Rods pick up light in our periphery and allow us to see black and white and shape and form, where cones are more in our central field of vision, and cones allow us to see color and detail. Now, once those rods and cones get stimulated, they in turn set off a chain reaction. That chain reaction starts with the rods and cones, but then will stimulate the cells that are next to them in the retina, called the bipolar cells. Those cells in turn will stimulate what's called the ganglion cells. Now, the ganglion cells are big, long neurons, basically, and they have long axons that wind themselves together to form what we would call the optic nerve, which then leaves the eye. So all of these are the axons of all these cells just wound together like a rope. And you can kind of see it here. That will eventually lead the eye and send the signal to the brain. But before we get to that, we need to talk a little bit about the optic nerve. When the optic nerve receives light, like you can see here, there are no rods and cones in the optic nerve. So light energy that hits that portion of the retina where there are no rods and cones, we don't see. And that is referred to as our blind spot. And sometimes our brain will actually just fill in the blind spot based on what it sees in the rest of the environment. So it kind of just fills stuff in based on what it's already looking at. And so the brain just does this naturally. And last week we did an activity where you tested your blind spot. So hopefully you were able to see that. From there, the signal goes to the thalamus, which is that train station of the brain, the message center of the brain. And finally, from there, it goes to the visual cortex, which is the area in the occipital lobe where we perceive vision. Now, within the occipital lobe and along with the visual cortex, we also have these things called feature detector cells. Feature detector cells allow us to detect certain features like shapes and form and connect it to prior experience so that we can identify it as things like faces and chairs and houses and so on. Now, it's another area that will recognize faces and connect it to individuals. But for everything else that we see and recognize, it's those feature detector cells that are picking up the shapes and form of the objects that we're seeing and then transmitting it and allowing us to kind of understand what it is based on prior experience. Finally, the last thing that we should be aware of is that a lot of people think that when the eye picks up light, it picks up a single image like a camera and then transfers that all the way to the brain where we see it. And that's not actually how it works. Instead, what's happening is that the eye is picking up each individual pixel of light. And all those pixels, those millions of pixels, are being sent to the brain all together, where then our brain then organizes all those pixels so that we can perceive what we're seeing. All right, the second part, and then finally, when we do see images, we see it in actually 4 distinct images that our brain kind of puts on top of, molds together, merges together. And these 4 things are color, motion, form, and depth. And this is what we would call parallel processing, in that our brain picks up 4 distinct images out of all those pixels of light and then kind of layers them so that we perceive what we're seeing. The next part of this is vision or hearing. I'm sorry. It's a little early for me. The next part of this is hearing. With hearing, just like when we pick up light energy, in this case with hearing, we're picking up sound waves. Now, sound waves are due to vibrations in the air caused by some sort of impact. Now, that impact could be your vocal cords creating an impact. It could be the impact of the air molecules inside a speaker headphone or an earbud, or it could be the impact that the speaker is making against the air. Eventually, those sound waves travel on air molecules in a 360-degree kind of ripple effect, kind of like if you throw a rock into a lake. That's kind of how sound waves work. And eventually, they reach your ear. Now, just like with light energy, we have 2 different measurements of sound waves that we can use to help understand it. The first is amplitude, and amplitude is the loudness, or it's kind of similar to the intensity of the light. So basically just how strong of a sound wave energy it is, and that will relate to the overall loudness. The other is its frequency. Now the frequency is basically kind of like at what type of sound wave is being sent into your ears. And the different frequency or the length of the sound wave will relate to the different pitches versus high sounds versus low sounds. When the sound wave hits your ear, it starts in the ear flap. That ear flap is specially designed to pick up frequencies that are associated with human speech. Then that sound wave travels down the auditory canal in the outer ear and will eventually begin to vibrate something called the tympanic membrane, but we would just call it the eardrum. That movement of the eardrum is based on the frequency and intensity of the sound wave. Now, that movement of that eardrum causes the movement of these 3 tiny bones in your middle ear called the hammer, the anvil, and the stirrup. And these three tiny bones are moving back and forth in unison based on the movement of that eardrum. Now, eventually, the third bone, the stirrup, is attached to a wound up snail shell looking like structure in the inner ear called the cochlea. The cochlea is filled with fluid, and when the movement of those tiny bones happens, it causes the movement in the cochlea, which then moves the fluid around in the cochlea, and that's where the key things happen with hearing. So if we take a look at it here, we can see kind of how these sound waves are traveling through the ear, how then the stirrup here might affect the cochlea, and eventually allow us to hear. Because in the cochlea, we have these things called the basilar membrane and the hair cells. Now, if we take a look back here, we can kind of see the basilar membrane and the hair cells are the inside of the cochlea. When you have the movement of this fluid in the cochlea, it causes the movement of the hair cells. And if you kind of think about like a coral reef that moves, where you have sea anemones moving according to the tide, that's kind of what our hair cells are doing inside our inner ears based on the movement of that fluid. Now, the basilar membrane is where these hair cells are attached to. And on the other side of this membrane, we have nerve cells. These nerve cells are auditory nerves. So the hair cells are basically being stimulated by the movement of the fluid, which creates electrical signals in the nerve cells that are on the other side of this membrane. So the membrane is kind of like a wall in the cochlea. And when these hair cells get stimulated, it creates electrical signals in the nerve cells. And then eventually those nerve cells wind themselves together, and we would call it the auditory nerve, sometimes called the cochlear nerve. This will eventually be sent to the thalamus, that train station of the brain, and then those signals will be sent to the auditory cortex in the temporal lobe, where we can perceive hearing. Just a little bit more about sound and hearing overall. Sound is measured in decibels. So decibels is the overall level of intensity of a sound. And decibels are exponentially higher. So something that's a decibel level of 0 is the minimum level of sound that we can hear. And yet 10, decibel level of 10 is actually 10 times higher than a decibel level of 0, rather than 10 units higher. So it's more exponential. When you get above 85 decibels, and I'm sorry, the picture is a little off in here, though I'll fix it for the actual lecture. When you get to 85 decibels, that can prolonged exposure above 85 decibels can produce hearing loss, where sometimes in some cases it can be permanent. When you have ringing of your ears that lasts more than, let's say, 5 minutes, that actually indicates bleeding of the inner ears, which is an indication of some level of permanent hearing loss, even if it's just a little bit. Finally, ultimately, one final thing is that hearing can be enhanced by our sense of vision. So kind of like both senses that we were talking about today, in a process called sensory interaction, where hearing is essentially amplified by vision. What ends up occurring is, let's say you're reading subtitles on a screen because the volume level is fairly low. Let's say it's quiet in your house because people are sleeping. That happens to me. When you look at the subtitles and you read them, it creates an expectation in your brain. That expectation in your brain allows your ears to become more sensitive to the frequencies associated with that expectation, or in this case, the words. And this allows you to actually hear the words better. And if you want to try that out, watch something at fairly low level where you can barely hear the dialogue, then put the subtitles on, and what you'll find is that you can hear it a lot better. And ultimately, that is overall the end of the lecture.